Hello, and welcome back to Introduction to Genetics and Evolution. I decided to add this extra short video because several people expressed that they didn't know a little bit about the scales of organization of genetic data. Now we talk about genes and we talk about how they affect a phenotype, and we talk about how they're hereditary. We mentioned DNA, but we never really said anything about how is it that the DNA sequence encoded in genes actually affects phenotypes. That could easily be the topic for an entire course. In fact, it is a topic for many courses. What I'll do is introduce just some very basic concepts just to help you understand the context of the class that we're doing right now on genetics and evolution. Now, how is genetic information translated to phenotype? Now, we've already talked about how DNA is the primary hereditary material. That is the material that is organized in chromosomes, that is passed from parents to offspring, that is allowing this mechanism for heredity. Now, there are sections within chromosomes that have specific instructions, and these sections are referred to as genes. This is a term that you hear all the time. Now, the question I'm posing to you now is how does this impact the phenotype? How is it the DNA sequence that's present in these different genes actually changes your hair color, or your eye color, or your personality, or your IQ? Well, a lot of this, not all of it, a lot of this is done through protein. Now, you hear the term proteins all the time in the context of your diet. Are you getting enough protein? Oh, this kind of food is high in protein. And in fact, proteins are vitally important to many, many aspects of your health, well-being, and development. Some proteins are structural. They're basically putting you together. Some are mechanical. They're essential proteins in various of your muscles, for example. Some of them are biochemical. The enzymes that you use for digesting your food, for example, are largely protein-based. And they can even affect things like cell signaling. So your hormones, for example, have protein components. Now, the central dogma of molecular biology, this is a fundamental principle. And this is an oversimplification, I should stress ahead of time, but it is nonetheless a fundamental principle of molecular biology, is that there are three primary levels of genetic information. They are also interrelated to each other. Now, DNA, as we've talked about, is that hereditary material. It is passed to offspring, but it also replicates itself, both inside your body and in the context of transmitting this genetic information to offspring. So DNA replicates itself. DNA also produces RNAs, and we'll focus on one particular kind of RNA here, referred to as messenger RNA. Messenger RNA is an intermediate between DNA and proteins. There are some forms of RNA that are directly functional, but again, we'll be emphasizing it in this video, the messenger RNA, those ones which are actually putting together the protein sequence. And finally, these messenger RNAs are translated into protein. Now, the structure and content of these proteins are what directly impacts your phenotype, or how you look, how you feel, how you act. Now, proteins are actually composed of chains of amino acids. Again, you've probably heard the term amino acids in the context of things you want to get particular kinds of in your diet. But if you look over here on the right, we have a string of amino acids. This would be part of a protein. If we zoom in on some of them, we see particular amino acids that you may have heard of: phenylalanine, leucine, serine, etc. Now, how is it we get from DNA to these proteins, aside from having this RNA intermediate? Let's talk about each step just very briefly. So the first step is DNA forming this RNA, specifically this messenger RNA. Well, the formation of messenger RNA from DNA is referred to as transcription. Okay? Now, as you may remember from other courses, DNA is double-stranded, and you have this complementary pairing. So for DNA to DNA, you always have these four different types: A, C, G, and T. These are the four different nucleotides of DNA. A always binds with T, C always binds with G, G always binds to C, and T always binds to A. And you see that over here. So here's DNA to DNA. A to T, A to T, T to A, C to G, G to C. Okay? Now, when you're producing the messenger RNA, you have the same sort of complementary relationship, except in this case, the T is replaced with a U. So this is a uracil. So in addition to being a different sort of compound, the DNA is deoxyribonucleic acid. RNA is just ribonucleic acid. It doesn't have the deoxy facet to it. You see it has the same sort of complementary pairing. So we have a double-stranded DNA that has been separated out, and we have this RNA transcript that is being produced in the middle, and you have the same sort of relationship of nucleotides. So you're essentially making a copy of the DNA as RNA. You know, the sequence here of this RNA transcript exactly matches the one here at the bottom, aside from this switch from T to U. See, TAC, TGCC, UAC, UGCC, etc. So this is how the DNA is making its copy as messenger RNA. Now what happens is this RNA copy of the gene sequence will leave the cell, leave the cell nucleus. It'll enter the cytoplasm, and that's where it directs the formation of these amino acids. So here, this messenger RNA, again, when it comes out, has to produce these amino acid chains. The term for this translation from messenger RNA to amino acid chains is translation. Essentially, we're taking this messenger RNA sequence and we're making it into a very specific string of amino acids. It's not just any amino acids, but it's very specific. And again, we have a code, just like the code we saw before. Now, this code for translation uses 3 nucleotides at a time, or 3 bases at a time. So this is a triplet code. So what happens, this is looking at the messenger RNA here on the bottom. We have an AUG, that is associated with one particular amino acid, methionine. ACG is associated with another amino acid. GUA is associated with another amino acid, etc. So it's going in this triplet code, 3 nucleotides at a time from messenger RNA to the resulting string of amino acids. Okay? This will continue until it gets to what's referred to as a stop codon. There are 3 different stop codons. This is one of them, UAA. UAA is not associated with amino acid, and it terminates the production of this string of amino acids. So we'll continue adding these on until you get to a stop codon, and then boom, done. We have completed the process. So let me show you how you actually identify these. There are tables, these are referred to as codon tables, that show you what AUG stands for, what UAA stands for in terms of amino acids. And here's a standard genetic code table. Here's AUG, as you saw before, is methionine. That is correct according to this table. UAA was the stop codon that you saw before as well. But you see all the different combinations. There's 64 different combinations because it's 4, since there's 4 nucleotides, to the 3rd power, because it's a triplet code. So 4 to the 3rd is 64, and that's the size of this table. There's 64 possibilities here, of which 61 are associated with amino acids. 3 of them are not. There's 3 stop codons that are not associated with amino acid. But we can immediately translate. CCC will give you a proline. An AAA in the messenger RNA will give you a lysine in the amino acid. So we have this perfect genetic code for putting these things together. Now, for closing, let me introduce some areas of the genome because I'll be using these terms occasionally and I want you to be familiar with them. Again, as we mentioned before, genes are these areas that have amino acid coding segments. Not all of the gene, however, codes for an amino acid. There are some segments of genes that don't. And one example of a segment of a gene that does not do so is the intron. Now, introns are areas within genes that are actually spliced out of the messenger RNA. So an RNA is produced from a large segment of the gene, but then these intron regions just essentially get cut out of the final messenger RNA as it's put together. As a result of that, they do not affect the final amino acid sequence. Okay? That's a very important point. So intron sequences do not affect the final amino acid sequence. The other region I want to introduce to you is what's referred to as intergenic regions. These are the areas that are found between genes. Now, just because it's not found in a protein-coding gene does not mean it's not important, does not mean it does not have a function. In fact, a lot of these intergenic regions are functional. Several of them regulate how much messenger RNA is produced, especially from neighboring genes, but not exclusively. And sometimes these intergenic regions even produce other kinds of RNAs that we haven't talked about here. So let me give you a fictitious example. These are 3 real genes, but I've diagrammed them, not how they truly are. So let's say there's the CHEK2 gene. CHEK2 you may have heard of because there's mutations in it that are associated with a 5-fold greater risk of breast cancer. I have another gene here referred to as NF2, another gene here referred to as SOX3. I've depicted them again fictitiously, so they're not actually right next to each other like this. So CHEK2, as I've depicted here, would have this whole segment which codes for a messenger RNA, and ultimately then is related to an amino acid sequence. There may be a segment here, this is the intergenic region, the area between it. This segment right here may actually affect, depending on the DNA sequence that's found here, may affect how much of this CHEK2 messenger RNA is actually produced. So this segment is actually vitally important, and it affects how much of a messenger RNA is produced, which will then also potentially affect how much of the protein is ultimately produced. So it does have a function, but its sequence does not affect the amino acid sequence of this CHEK2 protein. NF2, as I've depicted here, has two large regions that are associated with amino acid sequence, but also has an intron. So the messenger RNA that's ultimately produced from NF2 in this fictitious example would have this sequence attached to this sequence, but the middle part would not be present. Even though the middle part is there in the gene itself, it is not found in the messenger RNA. SOX10, similarly, has this large protein coding segment, this protein coding segment, this protein coding segment, but it has two introns. Again, those two introns would not be found in the final protein. And again, both of these may also have other sections here in the intergenic DNA that are regulating how much of the messenger RNA is produced of those two genes as well. Well, this was a whirlwind tour, but I just wanted to introduce some of these terms to you. I wanted you to understand the triplet code. I wanted you to understand the central dogma, this idea that DNA makes messenger RNA, which affects the amino acid sequence of proteins. And I want you to notice that the genes have these particular areas referred to as introns that do not affect the final amino acid sequence of protein. I hope that was helpful, and I hope it was interesting. Thank you for joining.
Hello, and welcome back to Introduction to Genetics and Evolution. In this video, I'll be talking about some basic processes for getting genetic material into new cells. So the two processes I'll be emphasizing in this mini video are mitosis and meiosis. I'm not going to talk about these in extensive detail. There's a lot of molecular parts to it, there's a lot of stages to it that I'm not really going to emphasize. I mostly just want you to understand these in the context of ploidy. Ploidy refers to the number of complete copies of genetic information in your cells. So you may remember in a previous video I referred to most animals being diploid, di-ploid. That di stands for 2. So most animals, just like you, have 2 copies of the complete genetic code in their cells. One that came from mom, one that came from dad, and that's why you're diploid. So let's talk about these two processes in the context of ploidy. The first process we'll discuss briefly is mitosis. Mitosis happens when you have the division of cells into 2 cells that are genetically identical to each other and to the parent cell that gave rise to them. So again, this is a process that produces 2 diploid daughter cells genetically identical to a single parent diploid cell. This happens inside your body all the time. When you're growing new skin cells, the genetic material is passed from the old cells into these new cells through the process of mitosis. So this is essential all the time through growth and development. Now, this also presumes that if you have no new mutations, we'll come back to this in just a second, if you have no new mutations, basically every cell in your body should be identical to the original fertilized egg that you were. So if you go back way back in time when you were originally one new fertilized egg, the genetic information in there, if it was not altered, would be true in all your current cells today. So let me ask the question, what would happen if we did have a mutation during mitosis? A mutation is a change in the genetic code. What would happen if that happened sometime during mitosis? Let's say when you were at the 16 cell stage, if one of the cells suddenly had some error in the genetic code where something changed moving on there. What would happen is you'd produce what's referred to as a genetic mosaic. And this shows you a picture of a flower that is a genetic mosaic, where there was a genetic change that happened in the cells leading to this half of the flower relative to those leading to that half of the flower. So there are different cells here that have a change in the genetic code from the others. Now, mutations in mitosis control genes have been known to be associated with various cancers. So this is often a very bad thing when it happens. So let's talk about the other process of splitting genetic information. Let's talk about meiosis. Meiosis, in contrast to mitosis, it produces haploid, or one genetic copy, daughter cells, from a parent diploid cell. Okay? So this is typically associated with the formation of gametes, such as when you are making sperm or eggs in your body. So you start off with all your cells having 2 copies of all genetic information, But then you basically lose one copy that then gets, and the other copy is what goes into your gamete. This is typically the precursor to fertilization. This is what allows it such that when you have kids, they also have two copies. Now, fertilization is what brings together the gametes of two parents into an offspring. So you have these single copies, these 1N meiotic gametes, coming together to make a new 2N, having two copies, diploid cell, which would be you, for example, or your kid. Now again, half the genetic material is from mom, half is from the dad. So we have this sort of union. And this picture diagrams it pretty well. Imagine this is a complement of chromosomes from dad. Here's a complement of chromosomes from mom. And you see that in the offspring, they have two copies of the blue, two copies of the red, two copies of the green, etc. Now, what would happen if you got two copies of a chromosome from one parent and then got a third copy from the other? What would happen if it didn't split up as much as it should? Well, the answer is not very good. You typically end up with some sort of fairly extreme disease. One of the most minor of these would be Down syndrome. This is when you have three copies of chromosome 21. Others are Klinefelter syndrome, etc. Often when this happens, the offspring is inviable and never makes it past a couple of cells large. Now let me give you a fictional example, and then I'll ask you a question after that to see if you've understood what I've said so far. Let's imagine that you're messing with your microwave. Microwaves are not as dangerous as I'm going to portray this to be. Let's say you're messing with this microwave. A lightning bolt strikes your house, and all of a sudden this big bolt of radiation shoots out of the microwave onto your face, and you grow a third eye. This would never actually happen. But let's say the cells in your face were affected. Now, these cells that have been changed in some way, would these cells spread by mitosis? Remember, they're in your head. Were they spread by mitosis? Could they spread by meiosis? And would you be likely to pass on the affected cells to your offspring or not? So I'm going to ask you these three questions, and we'll see if you understood from that. Well, welcome back. I hope you understood the answers to those questions. The answers, let me just briefly go over them. They could certainly spread by mitosis. Because again, mitosis is just what happens when your cells are making new copies of other cells. You're going 2N to 2N. In your head, you're probably replacing skin cells all the time, and they're making new ones. And if there's a mutated cell here, it might spread to other spots on your forehead right there. It is very improbable these cells would spread by meiosis because you're not usually making gametes in your head. You may think you are, but you're not. And because of that, it's also very unlikely you would ever pass on the affected cells to your offspring. Okay? So let's just recap real quick what's happening. So with mitosis, again, this is the process that's typically associated with just growth within your body. You start off in this fictional example here. There's only a single chromosome, but there's two copies of it. This is one that came from dad. There's one that came from mom. Before it actually replicates, before it actually passes on to the new cell, there is a period of replication. So you actually go through a very short 4N stage. We actually have 4 copies of everything. 2 from dad, 2 from mom. The 2 copies that are there, these are called chromatids. They are identical. It's just 2 copies of your mom's single copy, 2 copies of your dad's single copy. And eventually you get down to these daughter cells that are just like your original cell. These are identical to the original ones there. We have 1 copy of your dad's genes, 1 copy of your mom's genes. And it's just passed on over and over again. So mitosis, you start at 2N, you end at 2N, and you have exactly the same genetic complement in the beginning as in the end, assuming there's no new mutation. With meiosis, this is what's typically associated with gamete formation. Again, you start with this 2N, you start with the same 2N part, but here in this case, you actually produce 1N copies. So you are actually passing on only half of your genetic information to your kids. You're not passing on all the genetic information. And this works out such that the kid in the end has 2 copies because it has the one that came from you and it has the one that came from your partner. So 2N to 1N is what you typically see with meiosis. Now this is an important part. Meiosis is fundamental to understanding inheritance. So this is what we're going to focus on for most of the rest of the lecture. I hope you'll join us. Thank you.